tucked into the green folds of the Western Pyrenees, where Spain and France blur into a border neither government fully controls the way it controls the rest of its territory, there lives a people whose blood behaves differently than almost anyone else's on Earth. In some Basque communities, nearly a third of the population carries a blood trait that shows up in only about 15% of people worldwide. That number alone would be a curiosity. Layered on top of a language with no known relatives and a genetic signature that reaches further back than almost any other population in Europe, it becomes something closer to a genuine mystery, one that scientists have been chipping away at for the better part of a century without ever fully closing the case. Let's be upfront about how this video is framed, because the honest version of this story is more interesting than the exaggerated one. You may have seen headlines claiming an AI analysis cracked this mystery wide open. In reality, what actually exists is decades of solid genetic, linguistic, and medical research, cross-referenced and pattern-matched the way any large dataset gets analyzed today, computationally assisted but built on real fieldwork done by real geneticists and linguists going back generations. That research does reveal a striking pattern. It does not, however, hand us a tidy final answer, and any version of this story that claims otherwise is selling you certainty the data itself doesn't support. What follows is the real picture. What we actually know, what remains genuinely disputed, and why the disputed part might be the most interesting piece of all. To understand why the Basque people keep surfacing in genetics research, you first have to understand how unusual they are on several fronts simultaneously, because it's the overlap that makes them so hard to explain away as coincidence. Start with language. The Basques speak Euskara, and Euskara is unlike anything else spoken in Europe. Most European languages can be traced backward like branches on a family tree: Spanish, French, and Italian descending from Latin, English, German, and the Scandinavian languages sharing older common roots in Proto-Germanic, all of them eventually connecting to a larger Indo-European family that swept across the continent thousands of years ago. Euskara connects to nothing. Linguists call it a language isolate, a polite academic way of saying it has no identifiable parent language and no surviving relatives anywhere in the world. Some researchers believe it may predate the arrival of Indo-European languages entirely, a genuine echo of speech that existed in these valleys before the migrations that reshaped the rest of Europe's linguistic map. That alone makes the Basque people a linguistic anomaly. It is not, however, the only one. When geneticists examine Basque DNA, a similar pattern of distinctiveness shows up, this time written in biology rather than grammar. Most European populations today carry genetic signatures from several major waves of migration. Early hunter-gatherers, the farmers who spread out of the Near East roughly 8 to 10,000 years ago, and later pastoralist populations that moved in from the Eurasian steppe. That layering of migrations is, broadly speaking, the story of how modern Europeans came to be genetically who they are. The Basque population shows comparatively less of that later mixing. Multiple genetic studies over the past 2 decades have found that Basques retain a higher proportion of ancestry linked to Europe's pre-agricultural populations than most of their neighbors, consistent with a group that experienced somewhat more isolation from later migratory waves rather than one untouched by them entirely. It's an important nuance. Basques are not a people frozen in amber, unrelated to their neighbors. They're a population that appears to have absorbed less of certain later genetic input, likely because the mountainous geography of their homeland made large-scale population movement more difficult than it was on the open plains and river valleys elsewhere in Europe. And then there's the blood. Human blood is classified in several overlapping systems, but 2 matter most for this story. The first is the familiar ABO system: A, B, AB, and O. The second is the Rh factor, which comes down to the presence or absence of a specific protein, known as the D antigen, on the surface of red blood cells. If you have that protein, you're Rh positive. If you don't, you're Rh negative. Globally, roughly 85% of people are Rh positive, and about 15% are Rh negative, though that number varies significantly by region and ancestry. It's less common in many parts of Asia and Africa, and somewhat more common among populations of European descent. In the Basque region specifically, multiple independent studies conducted over decades have found Rh negative frequencies running well above the general European average, with figures commonly cited in the range of 25 to 35% depending on the specific community and study methodology. That places Basque populations among the highest documented concentrations of Rh-negative blood anywhere in the world. Basques also show an unusually high frequency of type O blood, in some communities exceeding 50% of the population, another marker researchers associate with older, less mixed lineages in European population genetics. None of this is a harmless footnote for hematologists to file away. Rh-negative status carries real medical consequences. It complicates blood transfusions, since Rh status has to be carefully matched or the recipient's immune system will react against the donated blood. And for most of human history, it quietly turned certain pregnancies into a slow, invisible danger that no one understood and no one could stop. We'll get to that history in detail, because it's arguably the most human part of this entire story. But first, it's worth sitting with the central unresolved question. Why does one relatively small, geographically isolated population carry this trait at such elevated rates compared to almost everywhere else? This is where the theories start reaching into deeper and more speculative territory, and where it's important to be honest about the difference between an intriguing hypothesis and a proven conclusion. A population that speaks an isolate language, carries an elevated share of pre-agricultural ancestry, and shows one of the highest concentrations of Rh-negative blood on the planet is not just a regional curiosity. It looks like it could be a genuine thread reaching back tens of thousands of years. Some researchers, and a great deal of popular science content online, have pushed that thread even further, raising the idea that the Rh-negative trait might connect not just to early modern humans, but to an entirely different human species that lived in Europe long before we arrived, the Neanderthals. For much of the 20th century, Neanderthals were popularly imagined as brutish, slow-witted, an evolutionary dead end that lost out to smarter newcomers and simply vanished. That caricature has been thoroughly dismantled by decades of archaeological and genetic research. We now know Neanderthals buried their dead with apparent ritual, made sophisticated tools, controlled fire, cared for injured members of their group, and survived punishing ice ages across a range stretching from Western Europe to Central Asia for hundreds of thousands of years, long before modern humans arrived on the continent. And when modern humans did arrive, the two populations didn't simply pass each other by. They met, and in some documented cases, they had children together. This is not speculation, it's written directly into the genome of most people alive today outside sub-Saharan Africa. Thanks to ancient DNA sequencing pioneered largely by paleogeneticist Svante Pääbo and his colleagues, we know that most people of European and Asian descent carry roughly 1 to 2% Neanderthal DNA, tiny fragments of an extinct human lineage still present in living people tens of thousands of years after the last Neanderthal died. Those fragments have been linked to real, measurable effects on things like skin pigmentation, immune response, and how certain bodies process specific compounds. Given all that, the question some researchers and science communicators have raised is whether the Rh-negative trait, or the broader unusual genetic profile of the Basque population, might trace back to that ancient Neanderthal admixture. It's an appealing idea. It would tie together the language isolate, the elevated ancient ancestry, and the unusually concentrated blood trait into one satisfying narrative about a population that held onto Europe's deepest genetic history more tightly than its neighbors. It's also, as of the most recent genetic literature, not something the hard evidence actually confirms. Large-scale studies mapping which specific genes in modern humans came from Neanderthal admixture have not identified the Rh-negative allele as one of the regions with elevated Neanderthal ancestry. The genetic architecture of Rh-negative status is well understood. It results from a deletion in the Rhd gene, and its distribution pattern across modern human populations is generally explained by researchers through founder effects and genetic drift within a population that experienced significant geographic isolation, not through a demonstrated Neanderthal source. The Neanderthal connection remains, at best, an unproven and largely unsupported hypothesis rather than an established finding, and it's worth being direct about that rather than letting a dramatic idea masquerade as settled science. What the evidence does support is more modest, but still genuinely fascinating. The Basque population, sheltered for millennia in a mountainous corner of the continent that later mass migration struggled to fully penetrate, appears to have preserved a genetic and linguistic profile that is measurably older and more distinct than that of most surrounding European populations. Whether the Rh-negative concentration specifically reflects a Paleolithic-era founder population that simply carried a higher starting frequency of the trait, amplified over thousands of years by genetic drift in a relatively small, isolated gene pool, remains the leading and most widely cited explanation among population geneticists, even if it's a less dramatic answer than an ancient species love story. Now to the part of this history that isn't really about ancient mysteries at all, but about ordinary families living through a form of grief that, for most of human history, had no name and no explanation. Picture a mother who is Rh-negative and a father who is Rh-positive, a pairing that is statistically unremarkable and occurs constantly across the world. If their child inherits the father's Rh-positive blood type, there are now, in effect, two different blood profiles present in what is temporarily one shared biological system. The mother, who lacks the D protein, and the developing baby, who carries it. Under normal circumstances, the placenta keeps the two blood supplies largely separate. That barrier isn't perfect though, and during the ordinary events of pregnancy, and especially during childbirth itself, small amounts of the baby's blood can cross into the mother's circulation. When that happens, the mother's immune system, doing precisely the job it evolved to do, encounters an unfamiliar protein and responds to it as a threat. It doesn't recognize a child. It recognizes an invader, and it begins building a defense. In most cases, the first pregnancy proceeds without any visible sign that something has shifted. The baby is born healthy. Life continues. But the mother's immune system, having encountered that foreign protein once, has done exactly what immune systems are built to do. It has learned, manufactured antibodies, and filed away a biological blueprint for a faster, stronger response the next time that protein appears. This process is now known as alloimmunization, and the cruelty of the mechanism lies in what happens next. The antibodies a sensitized mother produces are small enough to cross the placenta, unlike the baby's own larger blood cells. So if a second Rh-positive pregnancy occurs, the mother's immune system doesn't wait to see what happens. It sends its already prepared antibodies across the placental barrier, where they attack the developing baby's red blood cells, sometimes with severe and even fatal consequences, long before birth. For generations, this pattern played out in homes across Europe and well beyond it, and to the families living through it, it understandably felt less like a biological mechanism and more like an unexplainable curse. A healthy first child, followed by a second pregnancy that ended in loss, sometimes followed by a third that was worse still. Physicians of the era had no test to run and no protein to point to. Mothers were often told it was simply bad luck, or something wrong with their constitution, and encouraged to try again, frequently with the same devastating outcome. It's difficult to overstate what that repeated unexplained loss must have done to families who had no framework for understanding it, only the grim and mistaken sense that something about the mother herself was somehow responsible. The underlying mechanism didn't even have a formal name until the very end of the 1930s. Much of the credit for identifying that something specific and biological was at work traces back to a 1937 case at a hospital in Newark, New Jersey, where a physician named Philip Levine observed a mother suffer a severe transfusion reaction to blood from her own husband, blood that matched hers by every blood typing rule known at the time. It made no clinical sense under the existing framework, which led Levine and his colleagues to identify an additional, previously unrecognized factor in the blood, one that would soon be named the Rh factor after early research involving rhesus monkeys. Levine went on to connect this newly identified factor directly to the pattern of newborn deaths and pregnancy complications that had puzzled physicians for so long, laying essential groundwork that later researchers would build on. Giving the condition a name, hemolytic disease of the newborn or Rh disease, was itself a meaningful turning point. It transformed what had felt like scattered, inexplicable family tragedies into a single identifiable medical condition that could actually be studied, and eventually treated. By the 1940s, word of this discovery had begun reaching the public through newspapers, offering grieving families language for what they had experienced and the first real hope that a treatment might eventually exist. But for roughly 2 more decades, hope ran well ahead of the actual medicine. There was no injection, no procedure, nothing a physician could offer a sensitized mother beyond monitoring and, in later years, risky in utero interventions. The knowledge had arrived. The remedy had not. That gap began to close through the work of an Australian-born physician named John Gorman, working at Columbia Presbyterian Medical Center in New York alongside obstetrician Vincent Frieda, in close collaboration with immunologist William Pollack at Ortho Pharmaceutical. Gorman had been studying a phenomenon in immunology suggesting that, under the right conditions, the presence of existing antibodies could actually suppress the immune system's own response to a foreign protein rather than provoke it. Applied to Rh disease, the implication was almost paradoxical. What if you could prevent a mother's immune system from ever building its own arsenal against Rh-positive blood by introducing carefully prepared antibodies from an outside source, in the narrow window immediately after exposure, before her body had a chance to learn and remember? Testing that idea required deliberately exposing Rh-negative volunteers to Rh-positive blood cells and observing whether they developed an immune response, something that could never ethically be tested on women who might one day carry a pregnancy. So Gorman, Frieda, and Pollack's teams turned to Rh-negative male volunteers, most notably a group of inmates at Sing Sing Correctional Facility in New York during the early 1960s, an episode of medical history that would rightly be handled very differently under today's research ethics standards. Volunteers were injected with Rh-positive cells, the same exposure that triggers sensitization in a pregnant woman, and some also received the experimental antibody serum in that narrow protective window afterward. The men who received only the foreign cells developed antibodies exactly as expected. The men who also received the serum did not. Their immune systems never learned to recognize Rh-positive blood as a threat. The principle held. That finding became the foundation for Rho-D immune globulin, commercially known as Rogum, first approved for use in the late 1960s. Given to an Rh-negative mother within about 72 hours of delivering an Rh-positive baby, and often earlier in pregnancy as a further precaution, the treatment works by quietly clearing away any of the baby's blood cells that crossed into the mother's system before her immune system has the chance to build a lasting response. Within roughly a decade of its introduction, it was credited with preventing tens of thousands of infant deaths in the United States alone, and Time magazine named it one of the top medical achievements of the 1960s. In 1980, Gorman, Frieda, and Pollack shared the Albert Lasker Award, one of medicine's most prestigious honors, for the discovery. Today, hemolytic disease of the newborn, once a leading and largely unexplained cause of infant mortality, has been reduced to a rare, manageable condition in most parts of the developed world, thanks to a single carefully timed injection whose full mechanism scientists still don't entirely understand, even now. Which brings the two threads of this story back together. On one side sits real, well-documented medical history, a mysterious pattern of infant loss, a name finally given to it in the 1940s, and a genuinely elegant solution worked out through careful, if ethically complicated, experimentation in the decades that followed. On the other side sits a genuinely unresolved genetic mystery, a small population sheltered for thousands of years in the mountains along the Franco-Spanish border, speaking a language with no known relatives, carrying a disproportionate share of Europe's oldest ancestral genetic signatures, and showing one of the highest concentrations of Rh-negative blood found anywhere on the planet. The most dramatic explanation for that last fact, a direct genetic echo of Neanderthal ancestry, remains an unproven and largely unsupported idea rather than an established scientific conclusion. The more measured explanation, a founder population isolated long enough for genetic drift to amplify an old trait to unusual frequencies, is less cinematic, but it's the one that actually holds up against the current genetic evidence. What no one can honestly claim, at this point, is a fully settled answer. The concentration of Rh-negative blood among the Basque people is real, well-documented, and far too consistent across decades of independent research to be dismissed as noise. What produced it, and how far back its roots actually reach, remains an open question that continues to draw serious researchers back to those same green valleys, generation after generation, still trying to read a story written in blood that is, even now, only partly translated. So here's the honest question worth sitting with. Given a real and measurable pattern with no fully confirmed explanation, is the more responsible position to keep searching for a definitive cause, or to accept that some questions about deep human history may simply resist a clean, final answer? That's worth thinking about, and it's worth hearing what you make of it.